So we have completed with bacteria of kingdom Monera, and now we are moving on to Cyanobacteria. Cyanobacteria, also called as blue-green algae, not to be confused with green algae of kingdom Plantae. Okay, these are all are photosynthetic prokaryotes. These are all photosynthetic prokaryotes which are different from their photosynthetic bacterial brothers, in that photosynthetic bacteria had bacteriochlorophyll and these have a different set of pigments. The photosynthetic pigments that they have are chlorophyll A and phycobilins, which are also present in plants. Okay, how are cyanobacteria different from the photosynthetic bacteria that we saw under bacteria? Photosynthetic bacteria have bacteriochlorophylls, but cyanobacteria have chlorophyll A and phycobilins. Bacteriochlorophylls are not present in plants, but chlorophyll A and phycobilins are present in all the plants. Can we say that cyanobacteria could be primitive ancestors of higher plants? What was the second difference between cyanobacteria and photosynthetic bacteria? That the kind of photosynthesis that is carried out in photosynthetic bacteria is anoxygenic in nature. However, the photosynthesis that is carried out in case of cyanobacteria is oxygenic in nature. Oxygenic photosynthesis. This trait is also similar to plants. So two features are being similar to plants. So can we say that therefore cyanobacteria may have been ancestors of plants? Okay, they could be the ancestors of plants. Cell wall, however, is peptidoglycan. Why? Because they are prokaryotes, that's why. Because they are prokaryotes and plants are eukaryotes. Okay, prokaryotes are supposed to have peptidoglycan cell wall, not cellulose. Clear? Next, all are photosynthetic prokaryotes. Each and every member of cyanobacteria are photosynthetic in nature. Okay, let us look at them further. Okay, so this is a filamentous strand of cyanobacteria. Each cell in this strand is capable of independent existence. Okay, such kind of filamentous strand is called as a trichome. So here I have shown you a trichome. Some cyanobacteria are in the trichome nature, others are unicellulars. Okay, so in this trichome you see a number of cells connected to each other, and if you look closely you will find that there are two types of cells here. Some cells are thin walled, some cells are thin walled here, and some cells are thick walled. The thin walled cells are called as vegetative cells and the thick walled cells are called as heterocyst. Vegetative cells are thin walled and these are thick walled. Cyanobacteria carry out photosynthesis, right? Where? In the vegetative cells, sites of oxygenic photosynthesis. Clear? Then what are heterocysts involved in? Heterocysts are involved in nitrogen fixation. Nitrogen fixation. Okay? Plants can only perform photosynthesis no nitrogen fixation. Okay, and a bacteria like Rhizobium, which stays in the root nodules of leguminous plants, it can fix nitrogen but no photosynthesis. That's why it stays in symbiotic association with the roots of leguminous plants to draw nutrients, photosynthesize nutrients from the plant, because it can only fix nitrogen. All organisms require food source plus nitrogen. Okay, in animals we cannot do photosynthesis also, and neither can we fix nitrogen. We have to take both the sources from food. Plants can photosynthesize but no nitrogen fixation. Nitrogen they will have to derive from the soil, ya to symbiotic bacteria like these ones. Rhizobium can fix nitrogen but it requires photosynthesized nutrients. Okay, so each one is lacking something, as you can see, but these guys, Cyanobacteria, can fix nitrogen as well as photosynthesize. So they are said to be the most self-sufficient organisms. Okay, most self-sufficient organisms in biosphere. Why? Since can photosynthesize plus nitrogen fixation as well. Clear? Next, a mucilaginous sheath covers the trichome from outside. Mucilaginous sheath covers from outside, which protects from desiccation. Some examples of such kind of heterocystous cyanobacteria are heterocystous cyanobacteria, the ones having heterocyst. Examples are Anaerobic, nostoc, and there are some which are homocystous. Homocystous matlab, what do you think of homocystous? No heterocysts, only vegetative cells. Example is Oscillatoria. Clear? Habitat, they require mostly moist conditions, fresh water, marine. They can stay as symbionts, symbiotic relations, symbiotic relationships with lichens, coralloid roots, roots of cycas also house cyanobacteria. When we will be doing gymnosperms, that time we will discuss coralloid roots in some detail, and when we are doing lichens, that time we will elaborate on how the cyanobacteria affect this relationship. They can be unicellular, colonial or filamentous, and they are known to cause algal blooms. This phenomenon called algal blooms in polluted water bodies. So remember who is responsible for algal blooms over water bodies. Over water bodies which are polluted, which will have certain organic and inorganic nutrients. So these bacteria will spread on the surface by absorbing all these nutrients, and they will flourish. They will form thick mats on the surface of water which are called algal blooms. They might release toxins in the water and thereby affect the marine life of that water body. Okay, so algal blooms are not desired. So that is about cyanobacteria or eubacteria. The last eubacteria that we are going to do is Mycoplasma. Mycoplasma. Mycoplasma of eubacteria. Now you know prokaryotes are surrounded by a cell wall of peptidoglycan, isn't it? That's the characteristic feature of all prokaryotes except for this one. Okay, mycoplasmas do not have a cell wall. Okay, they do not have a cell wall. Okay, outermost layer will be plasma membrane. outermost layer will be plasma membrane, and as such they are resistant to certain antibiotics like penicillin. Why? Because penicillin is known to act on prokaryotic cell wall. If penicillin, an important antibiotic, acts on the cell wall of the bacteria and thereby brings about its disruption and thereby keep the infection under control by killing the bacteria by acting on their cell wall. What if you get an infection from Mycoplasma and you inject penicillin in order to kill the bacteria, but go to see the antibiotic is not working only on this bacteria? Why? Because it does not even have a cell wall. How will penicillin act? It wouldn't act. Okay, so they are said to be resistant to certain antibiotics like penicillin. Let us look at this structure. The outermost layer will be plasma membrane. Their structure is different from a typical prokaryotic cell in that this thing that is there scattered throughout the cytoplasm is its genetic material. How is it different from the nucleoid of a general bacteria? In general bacteria, the nucleoid is restricted to the center. The genetic material is coiled and restricted in the center of the cytoplasm. But over here, the DNA is scattered throughout the cytoplasm. Over there, it is circular. In a normal bacteria, the genetic material is circular in the center, but over here, it is linear. Both the ends of the genetic material are not close together. It's not circular. It is linear double-stranded DNA. It is linear double-stranded DNA that's scattered throughout cytoplasm. It's scattered throughout cytoplasm. Clear? At the start, at one point it will have this thing called as a replicating disc. Replicating disc for replication of DNA. Fine. Apart from that, you will see certain circular bodies inside. These are called as elementary bodies. Elementary bodies will have a copy of nucleoid and they can be released out from cell, and each elementary body is capable of forming a new cell around it using the information from the genetic material which is there inside. Okay, so they are involved in asexual reproduction. Apart from that, this will be the cytoplasm, and inside the cytoplasm the scattered ribosomes will be present, and since they are also prokaryotic cells, so they will be 70S type ribosomes, which is different from eukaryotic ribosomes which are 80S types. They will have 70S since they are prokaryotic cells. How are these bacteria different from the other bacteria that you have seen so far? They do not have a cell wall. Okay, and also they have a linear scattered nucleoid throughout the cytoplasm. What else? What else about the Mycoplasma? They can live without oxygen. Next, act as pathogens and cause diseases in plants, animals, and even humans. They can act as pathogens and cause diseases in plants, animals, and humans. They were first isolated from a diseased, they were first isolated from diseased cattle, okay, from their pleural fluids. You know what are pleural fluids? What is pleural fluid? These are lungs, no? Lungs. These lungs are covered by a membrane on the outside. This membrane is called pleura. Okay? Membrane is called pleura. And this pleura was filled with, this was infected by this guy, and a lot of pleural fluid was accumulated. So they were isolated from this, from the pleural fluids. And this disease that was caused was pleuropneumonia. And since they were isolated from here, they are therefore also called as pleuropneumonia like organisms, or PPLOs. They are also called as PPLOs. Mycoplasma are very popularly called as PPLOs, pleuropneumonia like organisms. And why are they called pleuropneumonia like organisms? Because they were first isolated from pleural fluids of cattle suffering from pleuropneumonia. Alright, so that is all about Mycoplasma. And with that we have completed Eubacteria part of kingdom Monera. We have completed bacteria. We saw bacterial cell structure, we saw bacterial nutrition, reproduction. That was about bacteria. Then we went over to Cyanobacteria. Then we completed Mycoplasma. These three aspects of Eubacteria have been completed. Thereby Eubacteria has been completed. Now we move on to Archaebacteria. Finally we are doing the Archaebacteria. Let us look at the Archaebacteria now. Archae means ancient. That's why they are also called ancient bacteria. They are also called as extremophiles. Phile means loving. So extreme condition loving. That's why extremophile, since they thrive in extreme conditions like hot springs, salty areas, etc. We're going to do three different types of Archaebacteria. Let us first start with Methanogens. Methano means methane, gen means generating. They generate methane, methane producing bacteria. Habitat, marshy areas, and in the gut of ruminant animals like cattle, which ruminate, okay, which keep on ruminating, chewing the cud. They will live in the gut of ruminant animals like cattle. And what will they do over here is they will oxidize CO2 and organic matter to methane, and therefore methane is produced in cow dung. Therefore cow dung will continuously emit methane, and that's why methane is a compulsory additive to biogas plants. Can you tell me why? Because biogas has mainly methane. Biogas is mainly methane, and in the biogas plants there will be organic waste and a lot of cow dung will be mixed together. Why? To supply these methanogens. Clear? They are said to be strict anaerobes. Strict anaerobes. There should be no contact with oxygen, which this condition they will find in marshy areas and in the gut of ruminant animals. Chemoautotrophic organism. They will synthesize their own food They will use the chemical energy from CO2 by oxidation of CO2 in an organic matter, and they will synthesize their own food. So that's about Methanogens. Write down one example. Methanobacterium. So that was about Methanogens. Moving on to the second type of Archaebacteria, that is Halophiles. Halophiles. Halo means salt. Phile means loving. Salt-loving bacteria. They are found in the extreme condition of high salt. Okay? High salt areas like Dead Sea, salt pans, etc. They are said to be photoheterotrophic. Heterotrophs. Photoheterotrophs. They will use light energy for ATP synthesis. They will use light energy for ATP synthesis, but do not use ATP for food synthesis. So food they will have to take from outside. That's why they are heterotrophs. Clear? They are said to have a pigment. Bacteriorhodopsin. Purple color pigment in their membrane for harnessing this light energy for ATP synthesis. What will they use this ATP then if not for food production? They will use this ATP that they harness from sunlight for their other cellular activities other than food production. ATP is required for so many cellular activities other than photosynthesis. Okay? So this bacteriorhodopsin pigment that is there in their membrane, which is a purple color pigment, it will impart a peculiar smell plus color to salt. Okay? Salt has a peculiar color and smell because of the presence of this pigment, which is called bacteriorhodopsin. One example, Halobacterium. Right? That is about halophiles. Moving on to the third type of Archaebacteria, that is Thermoacidophiles. Thermoacidophiles. Thermo means heat. Acido means acid. Philes means loving. Heat and acid loving bacteria. Okay? Habitat, hot water, sulfur springs. Hot water sulfur springs, wherein temperatures can be as high as 80 degrees Celsius and pH as low as 2, which is extremely acidic, but they will enjoy it. They will thrive in hot water and acidic conditions. What they will do is, they are basically chemoautotrophs. What will they do over there is that they will oxidize sulfur, which is there in the springs. They will oxidize sulfur to H2SO4, which is nothing but your sulfuric acid. This is the sulfuric acid, one of the very highly corrosive type of acid, and that is what lowers the pH of that water, but they will love it. Okay? And in this process, whatever energy that is liberated, this energy will be trapped into ATP, which they will use for food synthesis. So what are they? They are chemoautotrophs. An example of thermoacidophiles is Thermoplasma. So that was about Archaebacteria. Under Archaebacteria, we have completed Methanogens, Halophiles and Thermoacidophiles. And with that, we have completed with kingdom Monera, Eubacteria and Archaebacteria. Thank you.
महादेव के नाम पे सब्सक्राइब कर दें।